In this video, we're going to learn about something called dihybrid crosses. Again, you should know what characters are, traits are, what Punnett squares are. Review all that important terminology before you move on to this lesson. And again, we're focusing on inherited traits, which are traits that are passed on from generation to generation within the genes. So when we saw monohybrid crosses, we were studying only one character, for example, just flower color. And with monohybrid crosses, the genotypes only have two letters, like big A, little a. And then the gametes, they would have only one letter, like big A, or little a. A dihybrid cross, you probably guessed it, di means two, mono means one. A dihybrid cross is when you do a cross to predict more than one character at a time. So for example, I could be looking at flower color and height at the same time. So let's say flower color was represented by the letter A and height was represented by the letter B, then I'd have two alleles for flower color, because we're assuming still a diploid individual, right? You have to have one chromosome with the big A, one chromosome with little a, and then two alleles with the height characteristic on it. And so in this case, I am heterozygous for both flower color and for height, heterozygous. Notice for a dihybrid cross, the genotypes have four letters. And you probably guessed it, the genotypes for the gametes would have two letters. And I'm just going to show you an example of one. One of the gametes could be big A, big B. Another one could be big A, little b. So gametes always have half the letter, half the number of letters that the individual actually has. You could have trihybrid crosses, where you deal with three characters at a time. So it could be flower color, height, and P shape. And so for each of those characters, you have two alleles. Big A, little a for the flower color, big B, little b for the height, big C, little c for the P shape. And so for each of these characters, the individual here happens to be heterozygous. And as you guessed it, the gametes would have three letters. So an example would be big A, big B, big C. And I'll show you how to figure out the gametes for dihybrids and trihybrids and more later on. But right now, I just want you to get the idea that whenever you have the genotypes for an individual, the gametes have half the amount of letters, as you saw with monohybrids. That applies to dihybrids. Dihybrids, instead, you're dealing with two characters at a time, so that means you need to have four letters in your genotype and then two in your gametes. In trihybrids, you have three characters at a time, and that means you have to have six letters in your genotype and three in your gametes. And again, the reason it's two letters per character is because we are diploid individuals. For each character, we get one copy of a gene, one from mom. We get two copies of gene, one from mom and one from dad. And you could have more than trihybrid cross. You could have as many crosses as you have characters. So I could have a tetrahybrid cross with, that's right, you guessed it, 8 letters. I could have a pentahybrid cross or hexahybrid cross. I could have all these different crosses. What I want you to get is that the same concept applies. You have a certain number of letters in your genotype and then a certain number of letters in your gametes, half the number of letters. So you should be able to predict the number of letters in your genotype for any type of cross they do, dihybrid, monohybrid, trihybrid, so on and so forth. For diploid individuals, even triploid, that would be a great thinking question. And then figure out how many gametes they would have. Now, if the number of letters gets larger, your number of gamete combinations gets larger, that means that your Punnett squares also get larger the more that you add in the characters that you're studying. So for monohybrid crosses, we had a small Punnett square. It was a 2 by 2 square, meaning we had 4 squares total. For a dihybrid cross, we're going to have a 4 by 4 square. We're going to have 16 squares total, 4 by 4. And every time you add a letter, you probably guessed it, so rather than 4 by 4 for a trihybrid, we'd have an 8 by 8 for a trihybrid. An 8 by 8 for a trihybrid. And so if 8 by 8, if 4 by 4 is 16, 8 by 8 is 64 squares. So you can probably guess that it would be quite annoying to do Punnett squares for trihybrid crosses. That's why we're going to focus really on dihybrid crosses in this presentation. And then I'll teach you some tricks and tips to use probability and math instead of drawing Punnett squares, but that will be another lesson. We're going to learn again how to do dihybrid crosses. We'll learn how to predict the genotypes for dihybrid crosses and the genotype for the gametes, and then predict what the offspring will be. But it's the same idea. You draw a Punnett square, you do fertilization event in the boxes, and you figure out the genotypes and then the phenotypes of all the boxes. So we're dealing with more than one character. We're dealing with 2 characters at a time, and that's how we call it a dihybrid cross. So again, dihybrid cross, we keep track of 2 characters. Trihybrid cross, we keep track of 3 characters, and you could go beyond that. So here we have an example. We're kind of back to Mendel's experiment here. We're dealing with 2 characters this time, so you can see we have the color and the shape. So our color here is yellow, yellow, and our shape here is round, round, or smooth. And then over here we have our color is green, and over here we have wrinkled. And you can see that for the color, this individual is homozygous dominant. And for that texture, the individual is homozygous dominant. Over here, for the color, the individual is homozygous recessive. And over here, for the texture, the individual is homozygous recessive. So even though we deal with 2 at a time, you can still think of it as you're doing 2 monohybrid crosses at the same time. And then you can see the gametes that are possible here for this one over here would be big Y, little r. And the gametes that are possible for this one here are big Y, big R over here, and then little y, little r over here. And so to figure out the gametes, you need to realize that in a gamete, you're never going to have 2 of the same letters. You're never going to have 2 Y's in a gamete. You can have them in an individual, but not in the gamete. And that's because, remember, gametes form by meiosis, and these alleles for a given character are found on homologous chromosomes, right? So you have homologous chromosomes. I'm not going to draw them replicated just for the sake of simplicity here, but if you have a big Y on one of the homologs and a big Y on the other, remember that homologous chromosomes separate during meiosis. So that's why in gametes you're not going to have the 2 of an allele for a given gene in a gamete. You'll only have 1 of the Y's, 1 of the R's. And I'll show you that in more detail in just a bit. But these are the gametes that form for this one over here, and this is the gamete that forms for this one over here. Notice instead of 4 letters, there's 2 letters. For monohybrid crosses, we had only 1 letter in the gamete. So we'll see Mendel's experiment from a dihybrid perspective in just a bit. But again, dihybrid crosses, 2 characters. Trihybrid crosses, 3 characters. And again, like I said, you could treat each individual character like a monohybrid cross that you're doing. And I mentioned this already, that you have homozygous for the color here, and then homozygous for that texture there. So we have a yellow smooth, and over here we have a green wrinkled, homozygous for both characters. So let's say we had a different example. Let's say we had this one here, big Y, big R, little r. This individual will be yellow, so that's our color first because of that dominant allele. This individual here will be smooth as well because of that big R. So the individual is yellow and smooth on a phenotype level. Genotype, the individual is heterozygous for the color and heterozygous for that texture, as well. So again, in dihybrid, you're just dealing with 2 monohybrid just kind of clumped together. So let's take a look at why those alleles for a given character end up in different gametes. Here we can write down the genotype for our picture over here. So we have big G, little g, that's for one of the characters. And then the other one is big Y, little y, that's for the other character that we're looking at. And so we can go through meiosis here, and you'll notice that the homologous chromosomes separate. So the big G's will go in one cell, little g will go in the other cell. The big Y will go in one cell, and the little y will go in the other cell. And what that does is it results in our alleles being separated from each other, the alleles of a given gene. So you don't have big G with little g anymore, and you don't have any G's in the same gametes. So we end up with some gametes. We end up with big G, big Y, big G, big Y over here as well, little g, little y, and then little g, little y over here as well. And so what you may, actually I should draw them side by side. What you may have noticed is that that's not all the gametes that are possible. You could also have big G, little y, and little g, big Y. And you probably guessed it, based on this picture, on this metaphase plate, you could have had a different arrangement. Instead of having all the big letters on one side, I could have put this big Y over here and this little y over here. So the little y would have gone with the big G, and the big Y would have gone with the little g, and that would have given us our other set of gametes. So there's two things that we're seeing here again. We're seeing the law of segregation, which says that homologous chromosomes separate during gamete formation, during meiosis. So that's why big G or little g, or you can't have two G's in one gamete because the homologs separate, and the alleles for a given gene are found on homologous chromosomes, which are no longer in the same cell by the end of meiosis. The other rule we just saw here is the law of independent assortment, that you can have different arrangements about the metaphase plate, and that's why you don't just have to have big G and big Y and little g and little y. They can assort themselves independently and in different ways on that metaphase plate, and that's why you can get big G little y and little g big Y. You don't have to draw meiosis out every single time to figure out what your gametes are. We'll take a look at how to determine gametes in just a few minutes. So let's take a look at how to figure out what our gametes are. There's a few different methods that you can use. In monohybrid crosses, it was pretty straightforward. You just take the two letters that you have, big Y in one, little y in the other. So let's do the example of, we'll do our distribution method first. The distribution method I always find is the easiest one. So you take a look at what your gametes will have. So your gametes always need one of each type of letter in it. So let's start off with A. Let's start off with our big A. Some of the gametes will have big A, and some of the gametes will have little a. So we'll start off with the big A gametes. So big A could end up with a big B, depending on how it arranges itself on the metaphase plate. So this is one of the gametes. So big A is distributed to big B. Big A could also end up with little b. So we could have big A, little b, if the other arrangement happens. Now half the gametes will have little a instead. So little a will be with big B, or little a can be with little b. Little a, little b. The order of the letters, we just keep the same order that you see in the genotypes of the individual. You can also do there's another method I'll show you later on where you can make a little tree or a little flow chart with them, so that can work too. You can see this drawn out over here. I just prefer the distribution method. It's a little bit easier and you don't have to draw things out as much as you would for something this large and grand scale. So you should be able to use the distribution method or any method that you'd like to use for any of these genotypes that I have listed here, or any genotypes that you get. So I'll do one more example with you. I'll do the big T, little d. So big T, little d, little d. So some of the gametes will have big T, so you distribute that. You have big T, little d. And the other one's going to be the same, but I'm going to write it down. The other gamete's big T, little d, because there's only little d there. And then the other gametes will have little t, little d, little t, little d, little t, little d, little t, little d. So really there's only two types of gametes produced by this one, and the reason there's only two types of gametes is because you're homozygous for little d, so there's less variety that can be created there with your gametes because you have the same allele at the D locus. And again, you can do that for any one shown here. I just want to highlight that in the gametes you don't have repetition of letters. So I don't have two of any T's in there. I only have one T. And I only have one D in there because the homologous chromosomes have separated during gamete formation. That's our law of segregation. Now again, typically, the order of the letters, we try to keep it similar to what you see in the genotypes. Usually to determine genotypes, we go by the alphabetical order. So A would come first, B would come next, then C, D, so on and so forth. And then usually it's the uppercase letters that are written before the lowercase letters, if you have any lowercase letters in there. These rules that we just learned, they extend to any degree of hybrids that you do, any cross. So I could do the distribution method for a trihybrid individual here and figure out the gametes. So I could say that some of the gametes will have big A in it, and so it could be big A with big B with big D. Or it could be big A with big B again, but this time with little d. Or instead you could have big A with little b with big D. Or instead of with big D, it could be with little d. And then you can do the same thing, but this time you start off with little a. So it could be little a, big B, big D. It's the same variety, right? So instead of having my big A here, I'm just going to put little a everywhere. Little a, big B, little d, little a, little b, big D, and then little a, little b, little d. So you can see there's more variety possible for these trihybrid crosses. Then you can do the same thing though. Big A, if you had this example over here, you could do little a with big B with big D, and you can keep doing that distribution method every single time to get the number of gametes that would be possible in your crosses. Notice these are all heterozygous individuals here, so there's more variety of gametes than you would have for, let's say, a case like this where you have some homozygous individuals, because there's less combinations possible when you have two of the same letters that repeat themselves. So it's also thinking just logically as well. It's not always about memorizing the rules, it's about thinking logically about what are some possibilities in terms of the gametes that you have available to you. You can also do the branching method. So I'll show you this example with the branching method. Again, distribution in your head is a bit easier, so I could just do this in my head now. I know I have to have big A with big R, big A with little r, and then I'll restart with my little A with big R, and little a with little r, just like that. So just start off with just one of your characters and then see what they have to be combined with. If you want to do the tree method, there's a few different ways that you can do that. So you can have a branching method to show what your gametes need to be paired with. So I usually have my big A and my little a, I separate them, and then I show my big A could be with my big R or with my little r. My little a could be with my big R or with my little r. In reality, this is similar to the distribution method or in your head method. It's just more visual. So that would end up with A big R, big A big R, big A little r, and then little a big R, and little a little r, just like that. And so you should be able to do this for any genotype that's given to you, dihybrid, trihybrid, whatever hybrid, but we're going to focus on dihybrid. Here you can see the law of independent assortment that will come into play. I just want to illustrate that more visually. In this case, we have our big G, big Y, little y individual, and the chromosomes have arranged themselves with the big letters on one side and the little letters on the other side, which is why we ended up with the gametes big G, little g, big G, big Y, and little g, little y. But they could have arranged themselves differently and then obtained the other gametes that we don't see in this image. So they could have arranged themselves in this way here. So rather than have all the big letters on one side, we have big letter, small letter, big letter, and that gives us the resulting gametes that we did not see in the previous image. So we get our big G, big Y's, we get 2 of those. We get our little g, little y if that arrangement happened, 2 of those. And if the other arrangement happens, we get our big G, little y, and little g, big Y. And that's all because of our law of independent assortment. And as you guessed, there's more gametes possible when you have more characters that you're dealing with. So this is simplified. I mean, we have a lot more than just 2 characters in an organism, but we just simplify it and deal with 2 characters at a time. And again, you can see that image shown in this picture here. What I want you to get is where this happens, that idea shown in this picture here. I want you to get where all these different laws happen in meiosis. So for the law of independent assortment, remember that happens in metaphase 1 of meiosis 1. You can see that there's different arrangements possible. You could have the big letters all on one side and the little letters all on the other. Or you could have big letter, small letter, and that leads to different types of gametes depending on the situation. So if you go this method, you have those gametes there, and this method, you have those gametes. I don't want you to think that there's 8 gametes produced at a time. I just want you to get that this is one possibility that can happen, and this is the other possibility that can happen. The other idea I also want you to get in terms of why there's no longer the same letter within a gamete, that's because of the law of segregation. During anaphase 1, telophase 1, the homologous chromosomes separate. Big R will no longer be with the other R of its homolog partner because the homologs are now separated. So when I say what the physical basis means, that means when I say law of segregation, you have to explain what law of segregation means, that homologs separate, but when does that happen in meiosis? Well, you should know it's anaphase 1 of meiosis 1. And when I ask you about why there's so many different types of gametes that exist, yet we don't always see them in a picture, you should know that's because of the law of independent assortment, which happens about metaphase 1 because of the different arrangements that exist. So you can actually figure out how many different types of gametes exist using this formula here. 2 to the power of n, where n is the number of heterozygous characters. So it really has to be heterozygous characters because that means that you have different arrangements if you have heterozygous, if you have 2 different letters for each character. So if I have the same letter for each character, then I can't arrange them differently because it's the same letter. So over here I have big G, big Y, little y, the different types of gametes. Let's see how many are heterozygous. There's 1, 2. There's 2 heterozygous characters, so that means there's 4 different types of gametes, which you could see over here. 4 different types of gametes. Now if you look at this one over here, how many of my characters are heterozygous? Just 1. So 2 to the power of 1 would give me 2 different types of gametes that are possible. I'll make 4, but they'll be doubles. What about the GGYY? Well, let's see. Are there any heterozygous characters? No. 2 to the power of 0 is 1 type of gamete. And if you did the distribution method, you'd see you'd only get big G, big Y. There's only one way to arrange these on the metaphase plate because there's no heterozygous variety that's there. So when you have more heterozygous, you have more different types of gametes that are possible. When all your characters are homozygous, then you only have 1 possible arrangement about the metaphase plate, and that's why there's only 1 type of gamete that's provided to you. Alright, so now we're going to take a look at how to set up our dihybrid Punnett square and how to solve problems with dihybrid Punnett squares. So again, we're going to have a 4x4 matrix. That means 16 squares, as you can see here, 16 squares. Just like we saw with monohybrid crosses, you're going to put the gametes for one parent at the top. This time each gamete will have 2 letters, and then the gamete for the other parents on the side. And again, they're going to have 2 letters each. Then you could start doing the fertilization, just like we saw with monohybrid. We're going to have if you look at this example here, let's say we do little a big T with big A little t. Put your go in alphabetical order. So we're going to have a big A with a little a, which you can see here. Put the uppercase one first, and then a big T with a little t. You can see it over here. So we have big A, big T, little t. If we did this one over here, we'd have a big A with a little a. So we have that over here, but a little t with a little t. So big A, little t, little t. So in each of our squares, we're going to have dihybrid genotypes with 4 letters instead of having only 2 letters like we saw in the monohybrid crosses. But it's still the same idea, gametes at the top, gametes on the side from the parents, and then on the inside we have the fertilization event. If this is dad, this represents sperm. If this is mom, this represents the ovum or the egg. So it's figuring out what your gametes are, putting them in the Punnett square, and then figuring out what your individuals are. It does take a bit more time, but it's just as simple as the monohybrid crosses. So we'll do some examples from Mendel's experiments. So he first did pure breeding plants. He crossed the pure breeding yellow smooth with a pure breeding green wrinkled. And we'll do his Punnett square. So remember that you're going to have 4 gametes on the top, 4 gametes on the side. So we'll start off with figuring out the gametes for the RRYY. So there's only one possible gamete for it. It's the big R, big Y. I changed the order of the letters here. It doesn't really matter. And then the gametes for this one here, there's only 2 possible gametes for it using the distribution method, little r, little y. Now, in reality, I could have a much smaller Punnett square since there's not many gametes, but assuming that, let's just say 4 gametes are produced, I'm going to have big R, big Y here, big R, big Y over here, big R, big Y over here, big R, big Y over here, and then little r, little y, little r, little y, little r, little y, and little r, little y. And then I'm going to perform the fertilization. So I'm going to have big R, little y, little y. One more time here. Big R, big Y, little y. And you'll notice that you'll get that in every single square because our gametes are all the same. So in reality, I mean, I could have had only 1 square for this, but not all your crosses for dihybrid are going to be that simple. So you can do that for all these ones here, and you'd have the same result. And so remember that when Mendel crossed the pure breeding individuals, he ended up getting 100% of the dominant characteristics, and that's what we get. 100% of the individuals in these squares, they're going to be big R, so smooth, big Y, so yellow. The recessive phenotype seems to have disappeared for both. Another way you could write this is there's 16 squares, so 16 out of 16 would be big R, big Y, so smooth and yellow. And this is our F1 generation. So now we're going to have a larger Punnett square. Now we're going to take F1 generation and cross them together. So we're going to have actually different gametes, and it'll make sense to fill up a whole Punnett square for those different gametes. So here we have our heterozygous cross. We're crossing our heterozygous individuals with each other. So we have one parent over here, one parent over here. Let's figure out the gametes using the distribution method or the tree method, whichever one you want. So we have big R, big Y. So I'll write that up here, big R, big Y. The other parent is the same, is also heterozygous, so I'll do that at the same time here, big R, big Y. We have big R, little y is another possibility, right? Big R with little y here. Big R with little y. Big R with big Y. So big R, little y. Same thing here, big R, little y. Then we also have our little r, big Y, that's a possibility. So little r, big Y. Same thing for the other parent, little r, big Y. If you're not comfortable doing this all at the same time, just do one parent at a time. Do the top parent first, then the side parent after. And then, sorry, we had little r, little y as our last one. Little r, little y. And then little r, little y over here. And now we can do our fertilization event. So in this square, I'll have my big R, big Y, big Y. In this square, I'll have again big R, big Y, little y. I'm just gonna put a little dash here so I remember my little y. And I would do that just in case you can't tell your letters apart. Big R, little r, big Y, big Y. Big R, little big Y, little y. It is tedious, but it's pretty straightforward. Big R, big Y, little y. Big R, little y, little y. Big R, little r, big Y, little y. Big R, little y, little y. Big R, little r, big Y, big Y. Big R, little r, big Y, little y. Little r, big Y, big Y. Little r, big Y, little y. Big R, big Y, little y. Big R, little y, little y. Almost done. Little r, big Y, little y. And lastly, we have little r, little y, little y. And so that is our Punnett square for this heterozygous dihybrid cross. Now, just like we saw with the monohybrid cross, this is actually a really cool ratio here. Instead of having a 3-to-1 ratio, 3-to-1 was for monohybrid, which you can kind of see still existing in here. We have a 9:3:3:1 ratio. 9:3:3:1 ratio. This is our phenotypic ratio for the F2 generation in a dihybrid cross. So the F2 generation is the kids are of the F1 generation. And so what that means is 9 of the individuals are going to be dominant for both characters. Dominant phenotype for both characters. I don't mean dominant genotype, but dominant phenotype for both characters. Let's go take a look at that. So 9 of the individuals are going to be dominant for both characters. So what that means is 9 of the individuals are going to be yellow, because yellow is a dominant characteristic, and smooth. So let's go circle all the ones that are yellow and smooth, because smooth was the other dominant. So yellow. So whenever you have big Y, it's yellow, and big R, it's smooth. So here's 1, yellow and smooth. 2, yellow and smooth. 3, yellow and smooth. 4, yellow and smooth. 5, yellow and smooth. 6, yellow and smooth. 7, yellow and smooth. 8, yellow and smooth. 9, yellow and smooth. So there's our 9 yellow and smooth. We sometimes represent it like this. 9, let's say, big R dash big Y. Or I should say underscore. Because this here could be a little r or a big R based on the genotypes that we have. So in our 9-3-3-1 ratio, 9 of them are dominant for both characteristics at the same time, and we counted that here. So now let's take a look at what their other letters represent here. So let's do blue here. 3 of them are going to be dominant for one, but recessive for the other. So for example, 3 will be yellow, that's dominant color, and then are wrinkled, because that's recessive for the texture. So in other words, 3 will be little r, but then yellow, either big Y or big Y, little y. So let's go count that. Let's go see if that's true. So 1, wrinkled but yellow. 2, wrinkled but yellow. 3, wrinkled but yellow. And you probably guessed it. The other 3 will be dominant for the other and recessive for the other. So instead of being yellow, 3 will be green, because that's recessive for color. And at the same time, they'll be smooth, because that's dominant for texture. So it'd be like saying 3 big R, little r, big R, doesn't really matter, with a recessive for color, so little y, little y. Let's go take a look if that's true. So we have 1, smooth green, 2, smooth green, 3, smooth green, 3, smooth green. And so that is our 3 in our ratio. And yes, the last one is going to be one that's recessive for both. Green and wrinkled. Let's go take a look if that's the case. So that would basically mean a genotype of 1, little for both letters here. Little r, little y, little y. And that's what we have. Keep in mind this is out of 16. So each of these is out of 16. So 1 out of 16. And you can figure that out as a percentage. You can do 1 divided by 16 times 100. And that'll give you 6.25%. So this is all out of 16. All out of 16. Because we have 16 squares there. So this is our phenotypic ratio when you have a dihybrid cross for heterozygous individuals. 9-3-3-1. So in a multiple choice, if I ask you what percentage or what fraction of the offspring are going to be dominant for both characters, if you have a heterozygous cross, you don't need to do the entire Punnett square. If they're dominant for both characters, it's going to be 9 out of 16. So you can do your 9 divided by 16, and that gives you 56.25%. And if I said you have 100 individuals in the population, you take that 56.25% and multiply it by 100, and that means you get about 56, 57 individuals that are going to be that are going to have dominant phenotype for both characteristics. I could ask you similar types of questions for genotypes. So for example, what percentage of the population will be big R, big Y, big Y? So find that. Find how many times you have big R, big Y, big Y. So we have big R, big Y, big Y over here, 1. And it looks like you only have that 1 time. So you could do 1 divided by 16. Multiply that by, let's say you have, I don't know, 80 individuals in the population. So I'll do that times 80. And I get 5 individuals out of 80 that would be big R, big Y, big Y. So it's the same idea as monohybrid crosses, just you have more boxes to deal with, and you just need to be more careful and pay attention to detail because it's very easy to make an error here. I would strongly recommend you memorize this ratio, because on a test, if I don't tell you to make a Punnett square, but I ask you to show your work, you can use this ratio method as your work, and you don't have to spend 10 minutes making one of these Punnett squares. You could simply write down what the 9:3:1 ratio means, explain your answer using that, and within 2 or 3 minutes you're done. So you save 7 minutes on one question, maybe more like this, and that way you're done your test earlier. So that's your responsibility to remember this ratio, especially for multiple choice. So this is the ratio explained in some more detail. We have our 9:3:3:1 ratio. 9 individuals out of 16 will be dominant for both characteristics in terms of their phenotype, because this could be here a big A or a little a. This could be a big B or a little b here. Okay, 3 will be dominant for one, so let's say for the A, but recessive for the other. 3 will be recessive for the other, dominant for the other. And then 1 will be recessive for both characteristics. You can see here I put the genotypes for the 9 that are dominant. These are the genotypes, a big A, big B, big A, little a, so on and so forth, just like that. The 3 for the one that's dominant for A but recessive for trait B, you can see that here. So recessive, recessive, but dominant, dominant. Okay, there'd be 2 of these ones and 1 of these ones. How did I know that? How did I know without going back to count that I'd have 2 of these ones here, but only 1 of these ones? That's right, you probably guessed it. The heterozygous can form in 2 ways, right? Little a from mom, big A from dad, or big A from mom, little a from dad. 3 that are dominant for B but recessive for one trait, well, you can see that over here, these 2 here, and yes, you guessed it, probably 2 of these and 1 of this because of the heterozygous. And 1 that's recessive for all, and it's only that one that you see here. And you can probably even go back here and guess how many of each you would have for this one. Yes, you would probably only have 1 of this one, just like you'd have for the recessive. Let's take a look over here. You'd probably have more of these ones because you have a heterozygous here. You'd probably have 2 of these. Heterozygous here, 2 of these. That's 1, 2, 3, 4, 5. And then 4 of these because you have 2 heterozygous possibilities here. So you can guess which one would be more numerous in the population based on the number of heterozygous characteristics that you have, because there's more ways to produce those heterozygous individuals. And so you can see here that all we really did is treat these as separate monohybrid crosses, and we put them in one big square. Make sure you review this idea of law of independent assortment. It'll be very important to help you make your alleles, and it's also a very important connection to meiosis. That's why we studied meiosis at the beginning of the unit, because it does relate to this. A lot of students forget how it relates, but it does. All those gametes that you're studying and all that fertilization, it relates to everything we studied right at the beginning of this unit, the first half of the unit. Here we can see an example of what diversity would look like without law of independent assortment. So if you didn't have independent assortment, if your gametes had to arrange themselves in a certain way about metaphase 1, let's say you could only have big letters with big letters and little letters with little letters, then what would happen is your phenotypic ratio would be a 3:1 ratio for dihybrid crosses. I don't mean monohybrid, I mean dihybrid crosses, because you could only have big Y, big R, little y, little r, and when you combine those, you'd get 3 that are dominant for both and then 1 that's recessive. And so that was one of the hypotheses that scientists initially thought would happen. That was one of the main guesses they had. But then they also said, well, what if that's not the case? What if the big R doesn't have to be with the big Y? What if they could switch it up a bit? That's the other hypothesis. That was the hypothesis of independent assortment. And so they said, well, if it's dependent assortment, meaning if big Y has to be with big R, if the letters depend on each other, big and little, then you're going to get a 3:1. So if we get that result, we know it's dependent assortment. But if it's independent assortment, if the big letters don't have to be with little letters, then we're going to get that 9:3:1 ratio. And that's what scientists got when they did those crosses with plants. They got 9:3:1 ratios, which supported this idea here instead. So even though we can't see chromosomes, or we couldn't see chromosomes back then, we could use our knowledge of math to solve our hypotheses using visible things that we could see, colors and textures. So this was refuted. Refuted just means was wrong, not backed up by the data, not supported by the data. whereas this was supported by the data. That means it was backed up or was, let's say, right. I don't like to use the word proven, because at any point in time you could always have some experiment that disproves some hypothesis you have, but we say it was supported by the data in science. And so here you have some more sample problems that you can attempt. I would pause the video, try it yourself, and then check my answers after. So we have a situation here. We have in mice, the normal long tail phenotype is dominant to the short tail trait. A black coat color is dominant to the brown coat color. If two long-tailed black mice, heterozygous for both traits, are mated, what proportion of their offspring will be brown with short tails? So there's a lot going on here. Let's figure out, first of all, what the parents are. The parents are heterozygous for both traits. So we have long tail versus short tail. That's one of the character, I should say. So heterozygous for both characters. And then we have a brown coat versus a black coat. And the black coat is dominant. So let's just make black the letter B, and long is dominant. So long the letter L for the tail. And we know that both parents are heterozygous, so that means they're big B, little b, with big L, little l. And that would be both parents. So I'm going to put crossed with another heterozygous individual, big B, little b, big L, little l. And so we need to figure out what the gametes are for the parents. And so you could do your distribution method. So I'm just going to do one of the parents over here on the side here. So we have big B, big L, little l. So big B could go with big L. And I'll put that on the top over here, big B with big L, because we're making a Punnett square. Big B with little l, that's another possibility. Or we could do our little b here, little b with big L, little b with little l. And that would be the case for both parents. So then you can go along here and put your big B, big L for the other parent, big B, little l for the other parent, little b, big L for the other parent, and little b, little l for the other parent. Now let's just analyze their phenotypes. Since they're heterozygous, this parent would be black and have a long tail. This parent would be black and have a long tail, because they have the dominant allele for both the characters that we saw in the question. So we found the gametes, and then we have to go and make a Punnett square to figure out the fraction of kids that would be brown with short tails. So before I even do that, let me write down what the phenotype for brown with short tails is, or the genotype for brown with short tails is. Brown with short tail, that would be, so for the offspring, I'm just going to write that in a different color here. That would be little b, because brown is recessive, little l, little l. And so hopefully you've noticed this. If this is a multiple choice, you would have realized this was a heterozygous for both characters cross. That's her 9:3:3:1 ratio. Remember that 1 out of 16? That's recessive for both. So right here I could have found that 1 out of 16 individuals, just by knowing it's in my 9:3:3:1 ratio, would have been little b, little l, and I could have answered this part B immediately. I could have done 1 divided by 16. So I'm going to put that in brackets here, 1 over 16, multiplied by the total number of individuals in the population, or in the offspring population, 450. And that would have given me 28 or 29 individuals. Let's just say around 28 individuals. 28 individuals. So multiple choice, I would have saved so much time. Now if I asked you to do a Punnett square, here's how you would do it. But again, you could also explain, if I didn't ask you to do a Punnett square and I just said justify your answer, you could have done a Punnett square the long way, or you could have done the ratio and it would have saved you a lot more time. But let's just do this Punnett square over here. So back to my little purple color here. I'm going to do this quickly. So big B, big L, big B, big B, big L, little l, big B, little b, big L, big L, big B, little b, big L, little l. Keep going. Big B, big B. I'll do all the B's first here. Big B, big B. Big B, little b. Big B, little b. And then the L's next. Big L, little l. Little l, little l. Big L, little l. Little l, little l. Same idea here. Big B, little b. Big B, little b. Little b, little b. And then little b, little b. And then big L, big L. Big L, little l. Big L, big L. Big L, little l. And then one more time. Big B, little b. Big B, little b. Little b, little b. Little b, little b. And let's do our L's next. So little l, little l. There's our one that's recessive for both. Big L, little l here. Little l, little l. Big L, little l. And this is the one that we're interested in. If you look carefully, there's no other like it within the box. That's 1 out of 16. And then once you have that, you can go and do the work that we did over here. So I've included a few other practice problems that you can attempt over here. So you can see that we're crossing two heterozygous individuals, and you have to make your Punnett square. And yes, it is a little tedious, but it is pretty straightforward as long as you take the time to do it. In another lesson, we'll learn how to use probability rules to do this, and you'll get the same answers as if you were doing a Punnett square. More practice problems are included here. So over here we have a homozygous dominant for both traits crossed with a white smooth guinea pig. What are the expected phenotypes in a large litter? I'm not going to do this whole question, but I just want you to think about it logically speaking. You could use a Punnett square, but think about what the only thing you can get is. Your parent here, this one here can only give big B, and this parent here can only give little b. So right away we know we're going to have our big B, little b. This parent here can only give big R because there's only big R to give. Big R right there. And this parent can only give little r. Little r. So this is the only possibility of kids that you're going to have in this litter. You're going to have black and rough coat. That's it. That's 100% of your litter. You could do that in a Punnett square if I asked you. But if I didn't ask you to do that, you would have simply justified your answer the way I did. So within a sentence or two, you explained that your entire litter will have to be black and rough coat because the only allele this parent can give is big B, this parent can give little r, and this parent little b, and this parent this parent little b, and this parent little r over here. So by understanding the concept, you can save yourself time in multiple choice and even those short answer, long answer questions where I don't necessarily ask you to use a Punnett square. You will have to know how to do the Punnett square, but I want you to understand the concept more. And I'm just going to show you one more little shortcut as we do these Punnett squares. So here we have a cross between a homozygous or a heterozygous individual and a homozygous recessive. Yeah, that's right. This, you may have noticed, is a test cross. So you could actually figure out if your individual is homozygous or heterozygous for the characters in a dihybrid. And it's the same kind of result. If you end up with any of the recessive for the A, then phenotypes for the A, then you know you were heterozygous for A, and any recessive for the B, then you know you were heterozygous for B. But if you end up with dominant for one or for both, then you know you were homozygous for both. So it is a test cross. But just to show you how to do this Punnett square, like I said, Punnett squares are usually 4 by 4, so 16 for dihybrid. But take a look at this carefully. The gametes for big A, big B, little b are going to be big A, big B, big A, little b, little a, big B, little a, little b. And so you make your square and you put those gametes on top. Big A, big B, big A, little b, little a, big B, little a, little b. And then you figure out the gametes for your other parent. The other parent only has one type of gamete. So in reality, you don't really need to make your 4 by 4 Punnett square. To save time, just put that one type of gamete here. You don't have to repeat it. So our Punnett square will be big A, little a, big B, little b. Big A, little b, little b. Little a, big B, little b. And then little a, little b, little b. Just like that. And so you can see in this test cross we have the recessive showing up for both here. If the recessive shows up for both here, then you know that this individual here must have been heterozygous for both characters instead of homozygous for one or homozygous for both. But the idea is that it's still the same idea with fractions. So let's take a look at our genotypes here. So our genotypes, instead of saying out of 16, we're going to do out of 4. It's like a simplified fraction. So for the big A, big B, little b, we have 1 out of 4. But if you did your Punnett square, you would have had 4 out of 16. But it's the same fraction. It's the same percentage. For this one here, 1 out of 4. Or 4 out of 16 if you did your whole Punnett square. Over here, 1 out of 4 or 4 out of 16. And over here, 1 out of 4 or 4 out of 16. Let me just add in that word there, test cross. And again, we know this was a heterozygous individual for both because we ended up with this individual over here. I'm just going to do another example where let's say we had the dominant individual for the A's. We didn't know it was homozygous for the A's, for the dominant A, but we want to figure out if it was or not. And then we have our heterozygous for the one character there, and we want to do a test cross to figure out is it homozygous or homozygous for those characters. So we do our test cross, and then you can figure out your gametes in this case. So let's just look at our gametes here. We have big A with big B, and then big A with little b. And I could do the other ones. I could do big A with big B again, and then big A with little b again, but I'm not going to repeat the gametes. I'm just going to keep it a nice simple Punnett square. And then over here, only one type of gamete, so little a, little b. And I got big A, big B, little b, big A, little b, little b. And so here's what I want you to notice. The only character that we see the recessive in is the B. That means that our B must have been heterozygous. If I didn't know these gametes and these genotypes, the fact that I did a test cross and I got a recessive little b character means that my B must have been heterozygous. My A, I didn't see the recessive come back. So that means that this parent could have only given me big A as a possibility. And so I want you to think about this as you do it. It's not about memorizing, it's about understanding the concept. And if you feel more comfortable doing the 4 by 4 Punnett square, you can do it. You'll get the same results, except instead of getting 1 out of 2, you'll get 8 out of 16. 1 out of 2, 8 out of 16. So it's about developing that sense, or that general common sense or general understanding of what these numbers mean in that equivalent fraction. So here we have a few more videos that you can go over to practice some more dihybrid crosses, along with some more practice problems with the answers included here. In your textbook, there's some great tutorial pages. When you go through the readings, you'll see tutorials that show you how to do dihybrid crosses. So if you didn't like what I did in this video, check out this video here, check out other videos online, and check out your textbook tutorials. They are great explanations, step by step, of how to do each type of problem. You need the practice problems. You need to have experience reading the problems and interpreting them yourself. If you don't get it the first time, you don't have to go ask somebody immediately. Try to struggle a bit and try to figure it out for yourself. What does that mean? because in a test, you can't just always ask, what does that mean? You need to try and logic your way through the question, because things will be worded differently. Things will be worded differently on a test than in real life, and you gotta kind of figure out strategies to figure out what the question's trying to ask. Here are some more practice questions. One of the questions requires you to use table 14.1. I've included it on the next slide. This is from a different textbook. These what if questions are really good if you want to take biology in university. One of the textbooks you'll use, you can ask me to see it, is this big biology textbook, and usually they organize questions, and one of the questions that they have is a what if, where they make you think a little bit about the concept and apply your knowledge, not just regurgitate the information. So you can see that table on this page here, and again, it's just an explanation of those different characters and that 3-to-1 ratio when you have the heterozygous individuals eventually. And so what we learned in the monohybrid crosses was that for phenotypes you get our 3-to-1, 3 dominant for a character, 1 recessive for the other, and then for genotypic ratio it's our 1 big P to 2 big P to 1 little p when you're crossing heterozygous individuals with each other. So for example, big A little a with big A little a. So that's our first Mendelian ratio that we learned in this unit. Or those were our first 2 Mendelian ratios we learned in this unit. Our last Mendelian ratio that we learned in this unit, which was the one we did in this presentation, is our 9:3:3:1 phenotypic ratio. These are not genotypes, these are phenotypes. So 9 dominant for both, 3 dominant for one, recessive for the other, 3 dominant for the other, and recessive for the other, and then 1 recessive for both. And you should be able to tell me where these 9 come from, where these 3 here come from, where these 3 come from, and where this 1 over here comes from based on the Punnett square that you did or the Punnett square above. That shows true understanding. Again, these are not 9, 3, 3, and 1, but if you simplify the ratio, you'll notice it is a 9:3:3:1 ratio. So we didn't actually just have 16 plants or 16 peas. We had many more, but when you simplify the ratio for all those characters, we always got that 9:3:3:1 ratio as an end result. So for example, this 315 is 9 times bigger than that 32. This 315 is 3 times bigger than this 108. This 315 is 3 times bigger than that 101. So that's 9 to 3, 9 to 3, 9 to 1. So we always do the ratio of the first one to the other ones in there, and that's how we get that 9:3:3:1 ratio. And this is a summary of Mendel's model to explain all those ratios. And again, he didn't even know genes and chromosomes at the time, but these are all the different rules that we saw. So law of segregation to show why you don't have 2 of the same letters in the gamete. Law of independent assortment to show that the letters don't have to be big with big and small with small, and that's why you get so many different types of gametes. Principle of dominance to explain why sometimes a trait might appear to disappear, but it comes back in the next generation because it was recessive to the dominant one. He mentioned that we do inherit 2 alleles for each, I shouldn't say trait, I should say character here. That's a better word, because trait is just a variation of character. So sometimes if you see that, it's okay to question it for each character there. And the variation in traits is due to the different alleles that we inherit for those particular characters. So he really did use a lot of trials and a lot of repetitions and counting and math to come up with these ratios and to come up with all of these ideas, which today we see to be the case when we study chromosomes and know DNA in more detail. And again, this is simply amazing because he didn't know about DNA. He didn't really know about chromosomes at the time. He didn't know about genes. He just had this idea of these factors being passed along. These are the readings for this section, 210 to 212, practice problems on page 212, and do the tutorial pages. Those are super important. They give you the answers and the walkthrough and everything if you didn't like the explanation here. So go take a look at those. They will be very helpful. And then there's some practice problems on the PowerPoint and additional worksheets that you'll get to practice these questions.